Howdy, howdy. This video is going to look at extended conceptions of genetics that build onto basic Mendelian conceptions. So if you don't have a handle on Mendelian conceptions of genetics and the basics, you probably want to make sure you have that before you get into this one. Otherwise, it's probably not going to make a lot of sense. In this video, we're going to look at how Mendelian ratios are impacted by more complex modes of inheritance than just simple dominant recessive single allele kinds of situations. How the linkage of genes on chromosomes leads to non-Mendelian genetic ratios, and then how the environment as well can play a major role in affecting an organism's traits. Let's take a moment and let's get a handle on when Mendel applies. Mendel applies in what we would call simple inheritance. This is inheritance where there is a simple dominant recessive relationship for a particular trait that has two alleles, one that is the dominant and the other that is recessive. These traits have to be unlinked. If we're looking at multiple traits, they have to be on separate chromosomes, and it works in sexual reproduction. So, for instance, if we go to the dihybrid cross that we looked at in the basics, this works because each of these traits, seed color and seed shape, has a dominant allele and a recessive allele. And it works because the traits for seed color and seed shape are on different chromosomes. If we start to change that, we're going to change the ratios. We'll look at some examples to get a handle on this. A good example is a human blood type situation with A, B, and O blood types. This is a good example of an extension to Mendel's work because there are actually three alleles involved in this situation. You have the IA allele, you have the IB allele, and you have the lowercase I allele. The IA allele leads to what's called type A, the IB allele leads to what's called type B, and the lowercase I allele leads to what's called type O. So here we see a situation with three different alleles. And because there are three different alleles, there are now no longer three different combinations that you could have as possible genotypes. You now actually have 6 different possible combinations of genotypes. You can be IA, you can be IA, lowercase I, you can be IB, IB, you can be IB, lowercase I, you can be IA, IB, or I guess IBIA if you want to look at it that way, right? Or you could be lowercase I, lowercase I. Blood type's also a really good example of a situation where alleles are not in a simple dominance recessive relationship. Particularly IA and IB. IA and IB are co-dominant. What this means is that neither is dominant over the other, and so in the heterozygote, you'll express both. Both IA and IB are dominant over lowercase I. In order to understand how this works, let's talk about the actual makeup of blood cells. On the surface of blood cells, you have these antigens, this A antigen or B antigen. If you're type A, you have the protein, you have the gene to make the A antigen, which you express on the surface of your cells, and you can see it here as these little circles that are on the type A cells. If you have the IB allele, you make the B antigen, shown here as diamonds. But if you have an IA and an IB, you make both. And so as a result, you are type AB because you have both antigens present on your surface. If you're lowercase I, you don't make either. And this is why you're type O, because the O is like nothing, right? You're not making anything. But if you wind up having allele for IA and lowercase I, you're still going to express the A allele on your surface because you have one gene that makes that, even though the other one makes nothing. Same thing with B. And so as a result, you get this wonderful different variations that you can have in different blood types as a result, with these different combinations that are present. They also have pretty profound consequences for you as a person, because if you're type A, you can only get blood from people who are type A because you recognize that antigen as something that is not foreign to you, or type O because they have no antigen on their surface. If you get type B blood and you're type A, your immune system will treat it like a foreign invader and the blood will actually just clot in your veins. It will kill you. It's not a good thing. Same thing if you're type B. You can get blood from other people who are type B, and you can get blood from people who are type O, but you can't get blood from people who are type A. If you're AB, you can actually get blood from anybody, because your blood cells have both the A and B antigen on it. Your immune system recognizes A antigen as friendly, B antigen is friendly, and of course, O doesn't have any antigen, so it's not a big deal. If you're type O, you can only get blood from type O people because your immune system does not recognize the A antigen or the B antigen as anything other than foreign, and so we'll treat it as such. So we would say that type AB people are actually the universal receptor, whereas type O people are the universal donor. And so just by having one extra allele and a little bit of a difference in the relationships between how these alleles are inherited, we get a lot more combinations than what we saw in our simple Mendelian basic inheritance. Another good example of how Mendel's ratios can be impacted by different modes of inheritance is what's called epistasis. In epistasis, you have multiple genes controlling the production of a particular trait. A good example of this is coat color in mice. You have two alleles involved. You have the B allele and you have the C allele. Big B is going to give you a black coat, which is dominant to little b, which gives you a brown coat. C determines whether or not you make color at all. If you are big C, you will make color, and it is dominant to little c, which is no color. I don't like my little c. It's not little enough. Give me 1 second. Think about it this way. If you're going to have color, it works like this. Choice number 1 is being operated on by the C allele. And then once you get there, the next question is what color you're going to be. And that's operated on by the B allele. So if you don't make color at all, if you're little c, then you're not going to make any color. And so none of the rest of the chain even applies. You need to have at least one big C in order to then go into the consideration of what your coat color is going to be. As a result of this, you get different ratios. If you cross two heterozygote big B, big C, little c, who are both black, right? That's what they should be because they have one big C allele and they have one big B allele, so they're going to show that black phenotype. You're going to wind up with 3 different phenotypes instead of 4 different phenotypes, which is what we used to see in a dihybrid cross. We don't see that here. But also the ratio is different. You're going to get a 9:3:4 ratio of black to brown to white. And that's all because of the way that these genes are interacting with each other and the overall determination of what your color is going to be. So again, we see that changing the nature of the interactions from something different than just basic Mendelian inheritance has pretty interesting impacts on the overall distribution of alleles. Other examples of non-Mendelian situations involve linkage. Linkage refers to the connection of particular traits to particular chromosomes. Genes that are on a particular chromosome are linked to that chromosome. The first examples of linkage that were discovered were sex linkage, and they were discovered in fruit flies at the beginning of the 1900s. Fruit flies is another really good organism. They're very, very small. You can grow entire populations over 2 weeks in tiny jars full of food. Very, very simple organism to look at. The lab in which this work was being done was generating mutant fruit flies and then crossing them with each other to see what would happen. And they generated a white-eyed male mutant, which you can see here. And when they crossed that with red-eyed females, all of the offspring had red eyes. But when they crossed those with each other, they got back the white-eyed phenotype, but it only showed up in the males. In other words, it was connected to the biological sex of the fruit flies. Since then, we found all sorts of other examples of sex-linked traits, but these traits are being carried on the X chromosome in this case. In fruit flies, like in humans, if you're XX, you are going to be biologically female. If you're XY, you're going to be biologically male. The Y chromosome doesn't really have very much genetic material on it, but the X chromosome absolutely does because both biological sexes are going to have at least one X chromosome. So there are useful genes on there. And it just so happens that in this case, what you're seeing is a cross between females that don't have anything other than the normal red-eyed sort of condition on it, with male who has the white-eyed allele on it. When this moves throughout the generations, that white-eyed allele is going to travel with the X chromosome on that parental male. Linkage actually refers to a much broader phenomenon than just sex chromosomes, though it was first discovered with the sex chromosomes. It refers to any time genes are on the same chromosome. All of these genes for all of these different phenotypes in these flies are all linked to this particular chromosome. Each of these genes exists at a different place in the chromosome. And the only time that these genes will separate from each other is if crossing over during prophase 1 occurs in between the two locations of these genes. So the only time linked genes separate is if crossing over occurs. The further away the genes are, the more likely it is that crossing over will occur between them. And so the more likely it is that these genes will separate during meiosis. But generally speaking, if the genes are on the same chromosome, they're linked. And we should not expect that they will be inherited independently of each other. They'll be inherited together. There are also a variety of situations where Mendelian rules do not apply because the genes are not inherited in a Mendelian way. A classic example of this non-Mendelian inheritance is the inheritance of mitochondrial traits. Mitochondria have their own DNA, and all sexually reproducing organisms inherit their mitochondria from the maternal lineage, or from their biological mother's cells. The sperm does not deliver any mitochondria. So whatever traits are on the mitochondrial chromosome in your mother's egg cell is what you get, and in turn what you pass on. Crossing over, cell division, all of these things do not impact those traits, and so they are inherited matrilineally throughout time. In a cross between an unaffected father and an affected mother for a mitochondrial trait, all of the offspring will be affected because they're all getting their traits from their mother. Whereas if the father is affected and the mother is not, none of the offspring will have the trait because the father is not passing on the mitochondria to any of its offspring. This is a non-Mendelian situation. The other thing that we should point out is that there are plenty of situations where the environment has major impacts on traits. These two variations in coat color in the Arctic hare, for instance, are exactly genotypically identical. The coat color of the hare is entirely determined by the season and the time of year, where the white color is present in winter and the brown color is present in the summer. The hare will move between these two things. We, of course, see this in us, too. If we go out in the sun, we will tan, and our skin will darken as a result. There are a variety of traits that are influenced very strongly by the environment. And so it's really improper to think about any particular trait, particularly more complex traits like intelligence or height or weight or any of those things that we tend to think of when we think of traits as being solely under genetic control. Most traits are not solely under genetic control. They're under some combination of genetic and environmental influence. So as we wrap up this unit, we can return to our understanding of gene expression and expand on it. Certainly, the information in DNA is going to be transcribed into RNA, which is going to be translated into protein. And the action of protein is going to lead to traits. But it's not just the action of one protein. We have many other proteins as well that are giving rise to particular traits. Each of those is being determined through its own DNA. And the interactions of those proteins together is going to lead to a trait. But it's not just the proteins that are leading to the trait. Additionally, it's our environment. And so if we think of our DNA as the recipes for life, and the trait as the finished dish, it's probably best to think about the environment as the oven. You can't make the dish without the oven. In the same way, you're not going to have a particular trait if the environment isn't correct. And all of these other ingredients are present as well. Without those things, you're not going to have the thing that winds up being the physical organism. As we wrap up this lesson, let's check our learning with the following. Make sure that you can explain how more complex genetic interactions can impact phenotypic ratios. Make sure that you can use a more developed understanding of genetics to predict and explain the ratios of traits in the offspring of parents with particular genotypes and phenotypes, if you're told what that situation is. And make sure you can describe situations in which traits are influenced by more than just simple genetic inheritance. If you can do those things, you're doing great. If not, that's okay too. Take a moment, pause the video, write down any questions that you have so that you can get the answers that you need. Thanks again for watching. I really appreciate it. Have a great day.